Hi,
Hi,
I'm Ann,
and I'm Layla. I'm the LRN STEM for today.
and we're going to talk about mitosis. It's going to be like AP, it's going to be aligned with the AP curriculum. So we're just going to get started because I don't know when this meeting is going to end.
Let's see if it kicks us out.
Okay. Cell division.
Okay, so cell division is a process necessary in order for us to continue to grow, to reproduce. And one of these types of cell divisions that we're going to be covering in this video is mitosis, which is the division of somatic cells or your non-reproductive cells. And again, this is necessary if we want to reproduce and grow. Do you have
And
anything?
in multicellular organisms, it's for growth, repair. Like if you scrape your knee, then your cells divide to like repair that. And it's also for the development of fertilized cells, where a zygote, which is like the first cell that forms, it's from the sperm and the egg. Then the cells like continue to grow and become specialized. And cell division is in integral, right? Integral
Is integral?
in the cell cycle. Um, and cells need nutrients, and they also need to excrete waste through the processes. So it's like a surface, the volume of a cell increases faster than the surface area. So like the cell needs to divide into two to make sure that the surface area to volume ratio is, what is it? It's efficient for all of the cell processes.
Yeah, you have to have enough space in order to be able to do this stuff,
Yeah.
like we've been saying.
And okay, for the unicellular organisms, they also divide, but when they do cell division, it makes like a completely new organism because it's just one cell. So that's how they like reproduce. Okay.
Yeah, and in prokaryotic organisms, it can also be called binary fission. So
Like
that's just
bacteria.
yeah,
Because what they have linear chromosomes. So
yes,
it's it can't be counted as mitosis because of that.
exactly.
Chromosomes.
Okay,
Um,
chromosomes.
do you want to do the picture right now?
Yeah, I can draw.
Okay.
Not well, but I'm gonna draw. Oh,
Okay.
it says you cannot open a whiteboard during screen share.
Oh, do the annotation.
Okay,
Okay, I'll
yeah.
do it.
Okay, no, I figured it out.
Okay. Then I'll
Um,
undo this. Okay,
all
your
right.
turn.
So here we go. Here's our nucleus. And so what I'm going to draw in here is a chromosome. This is just one chromosome, 0 sister chromatids. And sister chromatids, what I'm going to draw here is when your chromosome duplicates. So in this cell, or we'll say nucleus number 1. And then here's number 2. So in number 1, we have one chromosome and 0 sister chromatids. And in number 2, we have one chromosome but 2 sister chromatids. And
How
in the
I
middle,
okay.
or sorry,
Sorry.
did I say something wrong?
No, I was just gonna say like how I think of it is like, yeah, if it has one centrosome, which is in the middle, that like circle that you drew, that like is how you know how many chromosomes there are. Because both of these have one
Okay.
chromosome, it's just the difference in the sister chromosomes.
Exactly. And so the centrosomes, we're going to go more into them later, but they are what join the 2 sister chromatids. And in parts of mitosis, you'll see that, like for example, in metaphase, where like chromosomes are lined up in the middle, mitotic spindles will attach to things around the centrosomes called kinetochores, and that's what helps pull the sister chromatids to the side of the cell, but we'll go more into that later.
Yeah. Okay. And also, hold on. I wanted to say something else, but I think I forgot it. Oh, well then there's somatic and then what is it? Somatic and gametes, which gametes are the reproductive and then
Yeah,
non-reproductive is the
so
somatic cells.
somatic is the types of cells that don't reproduce, are non-reproductive cells. And gametes, like Ryan said, it's like reproductive cells such as sperm and eggs.
I think that's it, right?
Okay. I cleared my annotations.
Okay. So the cell cycle, there's like two main parts, and here you can see like this big. Do you see my cursor?
Yeah, I can see it.
Okay, good. So this big part right here in like the light blue is all interphase because the cell cycle, a cell, it's like almost always in interphase. It's 90% of the time it's in interphase, and then the 10% of the time it's in mitosis, which is
Mitosis is the actual process of
of
sorry, what?
of like splitting.
Yeah, like it's the nucleus division, and then cytokinesis is the part where it's the cytoplasm division, but together mitosis and cytokinesis are the actual like physical cell division part.
Yeah. Is that it? That's kind of
Yeah, and then we can also, I don't think, and we have this stuff on the G1S and G2 on a different slide,
yeah,
but
about
as
like
you can,
the
yeah,
what is it, the checkpoints. Okay.
yeah,
So we
yeah,
can
we'll
move
talk
on.
about the checkpoints later.
So interphase, that's the first part. So oh actually hold on. In oh wait, yeah, never mind. Interphase. So that's like the 90% of the time, right? So there's three parts. There's G1, S, and then G2. S is the DNA synthesis where it actually duplicates the DNA, which like in Layla's picture, at first it was like 1 or 0 sister chromosomes, and then it was 2.
And then it actually like duplicated.
So, yeah, it duplicated. So it went from this to this.
That's good.
Thank you. Okay, so the nuclear membrane gets broken down, and then the chromosomes. Oh, hold on, that's never mind. That's prophase.
We're
I'll
almost
cut
there.
that out.
Not there yet. But yeah, so
Is there something else?
no, it's just basically these are the three subphases of interphase before we head into mitosis or the actual cell division. And like, okay, I'm just trying to like cover over everything we said.
And then, yeah, keep in mind in here, there's different checkpoints to check that the cell is actually doing what it's supposed to do in cell cycle regulation, but there's a slide for that later. And then, yeah, here's mitosis, which is after interphase.
Oh, and I
Prophase, metaphase, anaphase, telophase, cytokinesis.
And you can remember that using the mnemonic. I prepared meatballs after the cupcakes.
Okay. Oh, and here's a good picture because you're probably gonna have to like see a cell and like decide what stage it's in, like in Twilight, where they're in remember they're in biology, okay? Okay, they're in biology and like hold on, can I I'll show the Twilight video after we're done with this, but like if it's online, like Edward and Bella are like looking at a microscope and they're like metaphase, anaphase, and then
we did that.
Yeah, we did that, but like Edward was all impressed. He was like, "Wow, like you're so smart for a girl."
Oh.
Okay.
Women
So
in
here are the
STEM,
pictures. Yeah, because interphase, it's kind of like a clump of whatever, and you'll see in prophase it like condenses into like chromosome shapes.
Right. So in interphase you have your uncondensed.
Yeah. Okay. So this is like this is a better picture. So the nuclear envelope, you see it's like dashed because it's like starting to disappear.
It's like beginning to dissolve because I mean you can't have that as you're going through mitosis, the rest of the process of mitosis, because the chromosomes need to be able to move to different sides of the cell before splitting. So
and these centrioles too, plants don't have these, but they start to migrate to like the center to form poles, so that later on you'll see they'll form like these poles right here. They're migrating, and yeah, we'll talk about that later actually.
Later, we'll get there. We'll get there.
Yeah, we'll get there. And I think that's it, right? Okay, yeah.
Yeah, so again, like quick overview. The chromosomes are condensing here. They're like visible. You can see them in a microscope. the nuclear envelope begins to break apart, and your centrioles begin moving to the opposite cells, or poles, opposite poles.
Okay. Metaphase. What is this one? Meatballs. Meatballs.
Meatballs. Or middle. Meatballs are middle, but
I like meatballs, actually.
meatballs, yeah.
Yeah, okay. So in
So-
metaphase, the chromosomes, they line up in the center, as you
No,
see.
the middle.
The middle, in the meatball.
Mhm.
They align on the, here, this is kind of a good word, equatorial plane.
Okay.
They
It's
align
not
up
as good
on
as
the,
meatball or middle, but
yeah. You can either think meatball or equatorial plane, whichever you want.
or floats your boat.
Yep, and then these spindle fibers, they start to appear and they attach to the chromosomes.
So earlier, when I was talking about the kinetochores, this is what the mitotic spindles attach to. And mitotic spindles are made of the microtubules. So microtubules make up these mitotic spindles, which then attach to the area of the centrosome on a chromosome. And these are what help pull apart the sister chromatids in like anaphase or later. It's right after this, but it's still an important part of metaphase.
Okay.
And there is a checkpoint in metaphase where you have to make sure that the mitotic spindle is attached to the kinetochore correctly, but we'll cover that later.
Okay. Anaphase. This one's really pretty. Here, it already has a description. Let's read it actually.
Okay.
Or what is this one? In your meatball?
After.
Okay, after. This is
Or
after.
the way I think of it is apart because the sister chromatids are moving apart.
Okay, that's pretty good too.
Yeah.
So chromosomes separate at the centromere, and the sister chromatids move towards opposite ends of the cell by the spindle apparatus, while the polar microtubules lengthen, causing the cell to elongate.
Okay, that was a good description.
Thank you. Straight from my head.
Really?
Yep.
You're so smart.
Thank you. So, okay, let's play a game, okay?
Okay.
How many, what is it? How many chromosomes are in here now?
How many chromosomes? Okay, wait, let me count.
Yep.
10.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Wow.
That's good. Did I win?
Yeah, you won. Well, actually not yet. How many sister?
Okay, no. Now it's your turn. It's your turn
Oh,
to play.
okay. Sorry.
How
Okay.
many sister chromatids are in this picture?
0.
What? Why?
Because they're not attached to anything.
Oh. That's crazy. I had no idea. Good job. You won.
Okay, great. Is that it? Yeah, they're just pulled apart and then.
Yeah, so the newly split chromosomes, they move towards opposite poles of the cell. And I don't know, wait, can I annotate real quick?
Yeah.
Oh, I'm about to request. Wait, how do I? Uh-oh, did it kick me out?
No. Are you not allowed? Are you not allowed to annotate?
Okay, I can annotate now. That actually
Okay.
scared. Okay, so okay. So this right here, the thing I'm trying to draw, but it's not letting me. Okay, I'm so sorry. So this is a chromosome, one chromosome. However, if you're looking at this and this one, okay, that was supposed to be a star, and this one, these are stars. These were originally one chromosome together, two sister chromatids, but they were pulled apart, which is why they're now two individual chromosomes. They're not attached together anymore, so they're not sister chromatids anymore, but together they would have been.
Perfect. Oh, and also you can see as you can see right here, what I pulled straight from my head is they lengthen the microtubules here. The spindle fibers, they lengthen, which cause the cell to elongate. So this is where you start seeing like kind of not really distinct yet, but you see that the cell is pulling apart. Okay. Telophase.
Yay. This is V in my mnemonic, I think.
Oh, that's kind of boring.
Yeah.
Okay. Do you have something else like apart or middle for this one?
Do I have a what?
Do you have like for Metaphase, it was middle, and then for anaphase, it was apart. Do you have anything?
Okay, so this one, what I have in my drawing from ninth grade, wait, can you see? Can you see
Nope.
that? Light.
Oh yeah, yeah.
Okay. So what I have here is its take over.
Okay. Explain that. I don't really get it.
I wish I could. I don't remember like ninth grade. But so here in telophase, it's right before cytokinesis, and some part of cytokinesis does occur while the cell is going through telophase. So
Yeah, they're going like at the same time, right?
right, yeah. So in telophase, there are proteins that are in the center of the cell, and they pull the membrane inward, forming a cleavage furrow. And the cleavage furrow is what's visible in animal cells. Okay, that's an cytokinesis, but I'll like do a little bit on that, and it's basically your cleavage furrow forms, and like as that continues to form, the two cells can get pulled apart to form the two daughter cells.
Yeah,
And
because okay, cytokinesis, cytoplasm, easy to remember because that's where the cytoplasm is splitting. Like you can see the cleavage furrow here, and then the cell plate that eventually turns into a cell wall. But in telophase, it's the nuclei that are splitting, and then the
right.
membranes, the nuclear membranes in each one start to form again.
Right. And also your chromosomes are condensing here to form chromatin in this phase.
Okay. Yeah. And here, like the cleavage furrow, and then plants need a wall, obviously. So it starts from the middle, it starts forming in the middle, and then it goes out until eventually it splits.
Right. And I've just looked over something that I think I could have added to anaphase. It is something that does make sense if you're thinking about it and if you listen to what we said already. But so after your sister chromatids separate, so sister chromatids were the things that I drew with stars. Motor proteins can move these sister chromatids to the opposite ends of the cell. Like they can move them apart as well as with the microtubules. So
Oh, okay.
yeah.
Yeah, that makes sense.
Motor proteins.
Yeah, because motor proteins are on microtubules.
Exactly. And this is why the cell elongates. Like the cell elongates as this is happening.
Okay, perfect. Is that it?
That is it. That's all I have to add to that.
Okay, and now we're on to cell cycle regulation.
Yay.
I'm a little bit nervous for this one. We'll all
it.
be learning. We'll all be learning during this one, guys.
Right. No, we know our stuff. Come on.
Okay, let me actually look at my notes.
Okay, so there are signaling molecules that are present in the cytoplasm that can help regulate progress through the cell cycle. And it's molecularly based. Okay, and then here's some information that I found in the notes. Some of the initial evidence for this hypothesis came from experiments in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. So that can be connected to the cytoplasmic signals that are in the cell to help regulate them.
Okay.
Okay, so this is on cyclins and CDKs. I don't know when that slide is. Is that next or?
Yeah, but I kind of want to use this picture to
Yeah.
like, okay. So there's a G1 checkpoint, there's M checkpoint, and then there's G2, I guess. Yeah, this is G2, and then this one's
It's
M.
in between the two. So
What?
no, that's why it says like G2/M checkpoint. So it's
Oh,
like the
okay.
checkpoint that separates the two. You have to get past, like, for example, the G2 checkpoint in order to move into
Okay.
the
Yeah, that makes sense. Here, like you said it briefly, the M phase checkpoint ensures that the kinetic cores are connected to the spindle before it can move on.
Right.
And then what else?
Oh, in many cells, the G1 checkpoint, so that's the first one you're going to reach as you're going from G1 into the S phase. That is your most important checkpoint. It's a major checkpoint, and it's considered the restriction checkpoint in mammalian cells. If it receives a go-ahead signal at this point, then it's able to move on, and it can typically complete the cell cycle and continue to divide.
Okay. Here, also at the checkpoints at G1, S, and G2, if anything is wrong and the checkpoints detect it, then the cell like shuts down, or it can go into G0, which is kind of like a limbo where it doesn't divide, it just stays there until it like fixes itself or something. And then what else?
And how about apoptosis?
Do you want to talk about
Sure. So if the cell doesn't pass a checkpoint, it can do either of the two things that Rayanne had mentioned, or it can perform apoptosis, which is basically like a cell suicide. And all of your organelles and cell material, they're going to fragment, and then the cell is going to basically die. And it's not going to be, like, can't function anymore, obviously. Like, the reason why this happened was because it can't function as a normal cell does. So it shuts down, completes that, and then it isn't obviously able to go through the cell cycle because, it's dead. So,
Okay. Also G1 checkpoint is, it kind of is like, oh, is the cell allowed to start DNA synthesis? And then if it is, then it goes through. And then the G2 checkpoint, it asks if the DNA is copied correctly. And I think that's it.
okay. Yeah, I think that's pretty good. And then we're going to go more into it. And if we forgot anything, we can circle back.
Okay. Here, the CDKs and stuff.
CDKs and stuff.
Yep.
Yep.
Okay, I'm a little bit iffy on this.
Okay, so
can I start talking and then you can
I'll add on. Yeah.
Or correct me.
Or correct.
Okay, so there are okay, let me start off by reading this. The cell cycle is regulated by cyclin and CDKs, and CDKs are cyclin-dependent kinases, and the regulatory molecules of protein kinases and cyclins. And then protein kinases, they are used to activate or inactivate proteins with phosphorylation. And phosphorylation, that's like using ATP. And that's like all I got.
Okay, so I'm looking at these notes, going off of what you said. So particular protein kinases can give the go-ahead signals at your G1 and G2 phases. They can help you move forward, progress through the cell cycle. And because of the requirement for binding to a cyclin, this is why like the kinases are called cyclin-dependent kinases, the CDKs that Rayanne talked about. And peaks in the activity of one cyclin or CDK complex is called like an MPF, and they correspond to peaks in the cyclin concentration. We can go into MPFs a little bit. So MPFs are maturation promoting factors, or M-phase promoting factors, and they trigger the cell's passage past the G2 checkpoint to the M-phase.
To the M-phase, okay. I think we should just leave it at that, because then there's also cancer.
Yeah, cancer is important.
I also have here the regulatory proteins of cyclin and CDK, and MPF is like under CDK, isn't it?
MPF, yes.
So all of those are like internal factors that help the cell keep itself in control, but there's also external factors like growth factors, density dependent inhibition, and anchorage dependence. And growth factors are proteins that are released that stimulate other cells to divide. Sorry, this is my slime. And then density dependent inhibition, when if it's like in crowded cells, they just stop dividing. It's like a signal for them to stop dividing. Like this is helpful because if there's too many cells, like we'll get into like tumors and cancer, then they know to stop dividing. And then there's anchorage dependence, which these cells only divide with anchorage, they're anchorage dependent because they only divide when they're attached to something. So those are external factors that help with the control and the regulation of cell division. Are we good?
Good. Yeah, I think that's good.
Cancer.
Okay. So cancer cells have escaped from the cell cycle, the control of the cell cycle, and they're basically going crazy.
Yeah,
They
because
just divide really excessively and rapidly. And okay, what were you gonna say?
You can finish your sentence.
So that's basically it. They just
Okay.
excessively divide
Okay.
at like rapid rates.
Okay. I was gonna say like the internal and external factors that we talked about, they don't respond to them. So that's they don't respond to those cues that tell them to stop dividing. So they just keep dividing, and that's what gives you the tumors.
This is because cancer cells they manufacture or make their own growth factors. And just they have an abnormal cell cycle. Like it's altered from what a typical somatic cell cycle would be like. And cancer cells, their division is really random, rapid, but they can also just stop their division process at random points in the cell, or at random points in the cell cycle, sorry. And if they have a continuous supply of nutrients or food, then they can actually continue to divide continuously and indefinitely.
Okay. And there's proto-oncogenes. That's a gene that controls the cell division, and it encodes proteins that stimulate the cell division, and they're important for normal human development and for the maintenance of tissues and organs. But sometimes these proto-oncogenes can undergo a mutation and they become an oncogene, which are oncogenes can produce large amounts of growth proteins, resulting in excessive cell division, which is like the tumors. And when a cell can't regulate the rate of cell division, it turns into a cancer cell. And this can either be benign, which means it stays in its active site and it doesn't spread to other places, or it can be, benign and
metastatic, or not, sorry, I'm thinking of malignant goes to metastatic, but it's malignant, yeah.
Yeah, malignant, which means it like
I'm going to talk about
malignant is like dangerous and benign is like whatever. Like it, yeah.
Well, yeah. Okay, so metastatic cancer is basically cancer that originated in one area and it spread to a different area. These cells they undergo metastasis. I'm pretty sure that's how you say it. I mean, you can double check me on that. So cancer cells are exported to other sites where they can form secondary tumors, and this is how to all cancer spreads through the body. And you do have different treatments for metastatic. Oh, I can't pronounce anything today. Metastasizing cancers, such as high energy radiation or chemotherapy, which a lot of people have heard of. And these chemotherapeutic drugs, they interfere with specific steps in the cell cycle. And one of the examples in the notes is Taxol, which can prevent microtubule depolymerization, which prevents cells from going past metaphase. And if you remember, the microtubules are what attach. So microtubules are in the mitotic spindles, which attach to the sister chromosome or sister chromatids to pull them apart. And if this depolymerizes, if that's the word, then it's not able to progress past that stage. It can't continue to divide.
Okay. I also know that there are treatments that damage the DNA so that it can't duplicate and go on. So it just like what you said with the spindle fibers, it just attacks certain parts of the cell cycle to make the cells stop dividing. And also I have another thing for our oncogenes. Several genes can act as proto-oncogenes, so it takes 3 to 4 mutations to cause the harm to make it an oncogene, and then anything that can damage genes can cause harmful mutations, like carcinogens and mutagens. Okay.
Is that it for cancer?
Hold on. Mutations can be caused by radiation, smoking, pollution, chemicals, and viruses. I'm like, these are my notes from 9th grade, so I'm just gonna read them all. Like, what's the harm?
Stay away from that stuff. Not good stuff.
Yep.
Oh, and I have a fun fact, actually.
Okay, go.
So it's on chemotherapy. I took AP Psych last year, and it was a really fun class. And so one of the things that we learned was that when patients are undergoing chemotherapy, right before the beginning of their treatment, what was done in the past was they'd get, oh my gosh, we have 10 minutes left in the meeting.
Go, go, go!
So what they do in the past was they'd give the patient something that they really enjoyed eating. Like, for example, if you really liked strawberry ice cream, they'd give that to you right before you underwent chemotherapy. However, after chemotherapy, they ended up absolutely hating strawberry ice cream, even though it was their favorite food to begin with.
Oh, wow.
Yeah.
Why?
Just because they associated strawberry ice cream with the side effects of the chemotherapy.
Oh,
And that they weren't positive side effects.
yeah.
So that's why they did that. So
Okay, I have a
eat
little
something
bit more.
nasty.
I have a little bit more on the development of cancer.
Go for
So
it.
it develops after cell, and number one, it experiences unlimited growth, and because it turns on growth promoter genes. Number two, it ignores the checkpoints. So it turns off the tumor suppressor genes. And number three, it escapes apoptosis, and it turns off suicide genes, and immortality equals unlimited divisions.
Wow.
Got a little equation for you guys.
That was good. I love equations.
Thank you.
Just kidding.
Okay. Oh, and then, like I said, benign tumors remain at the original site, and malignant tumors invade surrounding tissues and metastasize. That word is highlighted. Remember it.
It's a word I can't say.
Metastasize. Oh, you already covered this?
Yeah, a little bit.
Okay. Well, it's a recap.
Go over it again. Go over it again.
Yeah. So metastasize is a process that the malignant tumors they go through to form additional tumors in places that they didn't start in, not in the active site.
Right.
I think that's it.
That was good. I think that was pretty good. I'm just going through the Campbell's textbook, making sure we covered everything. And
Okay.
I think we did a good job.
Let me actually see if I can make. Hold on. Let me just try something, okay? Let me try
Okay.
something. I'm gonna
Okay.
stop the share, and
Oh.
I'm gonna see if I can connect my phone here so that we can watch my
Your Twilight video.
stop motion.
Oh,
Oh no, not
what about
Twilight
the
yet.
Twilight video?
Here.
Got me excited for that.
Like we already studied, we can chill now.
You can enjoy
How do we
your
do this,
wonderful
actually?
drawing.
I used to do this all the time, and I used to watch movies on Zoom, but I kind of forgot
I
how.
haven't been on Zoom in such a long time.
I know. I forgot how to do it. Is it share screen?
Yes.
Oh
Sure.
yeah, iPhone, iPad. Okay.
Ooh.
Tap screen. Okay. Oh, okay. I'm excited.
Uh-oh, me too. I can't see your screen, but I'm
Okay,
excited.
you can see it.
Okay, now I can. Let's go.
Okay, perfect.
Show us
Here, we can.
sideways.
Okay, yes, I can't. Yeah, I can't switch it. Might
Okay,
be a
but
little bit laggy.
this is your cell cycle.
Yeah, but it doesn't have cytokinesis, but it's fine. You can imagine it. Just
Cytokinesis
remember?
is basically, remember cleavage furrow or cell plate,
Mhm.
and it's when your cytoplasm is dividing.
Cytokinesis, cytoplasm. Okay, I'm gonna play this for you guys.
Okay, let's see it. Whoa! The paper's
See the
getting
spindle
smaller.
fibers? Okay. Okay, perfect. Lining up in the middle. Now it's anaphase, telophase. Wow, perfect. Okay, now we can go slow-mo now. We can talk through this. So
Okay.
interphase number one, right? It's a mess. It's a mess. It's like completely a mess. And what are these things called? I forgot.
Are those your centrioles?
Are they?
Sorry, I'm just trying to fix that light. That light.
Okay.
Yes.
Okay. Okay, so your centrioles.
Okay.
They're right here. They're like stagnant. And then you can see now we're going into mitosis. Oh, but also remember in interphase it's like the three parts, right? G1, S, and G2. And in S, it duplicates the DNA. So now you go into mitosis.
Which is the division of the nucleus.
Yep, division of the nucleus. So number one, prophase, it starts to condense. Here I'm like animating it, okay? So at first it's like a blob, right? And now, hold on, they're condensing. Now they're
What about the
condensing.
nuclear envelope?
Yep, and now you can see they're individual, and then the nuclear envelope disappears. It's a little bit sudden because this is kind of my third stop motion, so I'm a little bit of an amateur. Okay. So now your centrioles are moving to the poles, right? And now you can see the spindle fibers. And here in metaphase, your chromosomes are going to line up in the center. In the middle, sorry, in the meatball. And now they attach to the middle. What is it? The middle quadrant?
What?
Kinetochores. The kinetochores are like, it's not the centrosome, it's like the whole middle part. It's not just the centrosome.
And also, just clarifying, the things that you drew on either end of the cells, those are the asters, right?
Yeah.
Okay. So yeah, what I'm pretty sure those things that ran, wait, am I allowed to annotate? Yes, I can. This, they're asters, I believe.
I thought those were the centrosomes.
I think that,
wait, not the
centrosomes are in the chromosomes.
No, not the centrosomes. What did you call them? Centrioles.
Centrioles, yeah, the centrioles form the aster, I'm pretty sure.
Okay.
So I think collectively it is called an aster. I did see that somewhere.
No, I did see it too. I'm just a little bit confused in the naming. Okay. Anaphase. That's when they get pulled apart. You can see their
naming. I want to cover over a little bit of naming, like once you're completely done going over this, because we do have 3 minutes.
Yeah, we have 3 minutes, and maybe we'll see if there's time for Twilight. I'll edit it in, actually. That can be our outro.
Oh, okay. Yeah.
Okay. And now in telophase, you see they've reached the end of their spindle fibers, and their nuclear membrane formed again.
Yeah,
and you see how there's instead of 2 chromosomes, now there are how many?
I can't see, it's blurry.
1, 2, 3, 4. 4 chromosomes.
Yay!
And 0 sister chromatids. So that's telophase. And then they unwind again, and they're all messy. And then cytokinesis, just imagine that there's the cleavage furrow that splits it up. And I have your 2 cells. Perfect.
Cleavage furrow in animal cells. Cell plate in plant.
Yep. Okay. Did you want to do the naming?
Right. So,
okay. Do you want me to show a particular slide?
I don't think there's a slide in particular. It's just like basic stuff that we may not have completely covered.
Okay. Go. 1 minute.
Oh my gosh. Okay. No, we have 2 minutes.
Go!
Oh my gosh. Okay. So all the DNA in a cell makes up the cell's genome. And the genome can consist of a single DNA molecule, which is common in prokaryotic cells, or a number of DNA molecules, which is common in eukaryotic cells. Eukaryotic are the multicellular organisms like plants, animals, things like that. And DNA molecules in a cell are packaged into chromosomes. We covered chromosomes a lot in this video. And they're the things that I drew, that Ryan drew, like they're just everywhere in the video. And eukaryotic chromosomes consist of chromatin, which is a complex of DNA and a protein called histones that condense during cell division.
So the DNA wraps around the histones to make chromatin, right?
Yeah, and all of that spirals and it forms into a chromosome. Okay, so I think that other than somatic cells being non-reproductive and gametes being reproductive cells, that's all the vocab that we need.
I think we're good. Less than 1 minute. Bye.
Oh, okay. Bye.
Everyone, good luck on your quiz if you are in Dr. Romano's.
Yay. We got this, guys.
See you tomorrow.
See you.
And on Friday, let's do Stripes Friday.
We don't have school Friday.
Oh my God. Stripes Thursday.
Stripes Thursday.
Okay. Spread the word. Bye.
Bye.
Mwah. Hold on, let me end it. Okay. Bye.
Yeah, maybe end it.